Hello and welcome everyone. In today's video, we're going to talk about receptors as a drug targets. Receptors are protein that mostly membrane bound, or in other words, they are integral proteins, containing a specific binding site for chemical messengers, which can be an endogenous molecules, those our body produces, like the hormones such as insulin, and neurotransmitters like noradrenaline, and exogenous molecules like the drugs. In a complex organisms like a human, there has to be a communication system between the cells. Imagine a heart where each muscle cell contracts at a different rate. It will be pointless and useless in its function as a pump. Therefore, communication is essential to ensure all organs and tissues operate in a coordinated and controlled fashion. Control and communication come primarily from the brain and the spinal column, the CNS, which receives and sends messages throughout the body via a network of nerves. At cellular level, once the message reaches the last neuron, there must be a specific way of carrying the signal from the nerve ending to the target cell. Here comes the role of the endogenous neurotransmitter we just mentioned and their receptors. The nerve releases this chemical messenger, which then diffuses across the gap to the target cell, where it binds and interacts with the receptor embedded in their cell membrane. This binding leads to a cascade of secondary effects, which results in either a flow of ions across the membrane or activating or deactivating an enzyme. Inside the target cell, a biological response is produced thereafter. If, for example, we are talking about the heart again, then the biological response might be the contraction of the muscle cell in a coordinated manner. So far, we have talked about the importance of receptors in cellular communication involving neurotransmitters. But cells also communicate through the other endogenous molecule, which is hormones. Once again, receptors are responsible for binding these circulating hormones and triggering an effect. But how drugs, the exogenous molecules, interfere with this communication system? It happens when the system gets messed up. It's clear that for a proper workings of the body, the communication must be normal. But when it goes wrong, huge variety of ailments can arise, such as depression, heart problems, muscle fatigue, and the list goes on. So what sort of things could go wrong in the system? It would be that too many or too few messengers are being released. At this point, the drug can play a role by either acting as a replacement of these messengers or by blocking the receptors from receiving their natural messenger to return everything back to its balanced state, the homeostasis. There are four main families of receptors in the body, which are ligand-gated ion channel receptors, G-protein coupled receptors, kinase-linked receptors, and receptors linked to gene transcription, or the nuclear receptors. They differ in the structure and function. Now let's see how each receptor of these works. Starting with the ligand-gated ion channel receptors, also known as the ionotropic receptors. The movement of ions like potassium and sodium across the membrane is crucial for nerve function and communication. But ions are charged molecules, so they can't pass the membrane through simple diffusion. It rather moves through these hydrophilic channels, but there has to be some control, like a lock gate that opens and closes when required. This lock gate is controlled by a receptor protein sensitive to external chemical messenger, where the receptor is an integral part of the ion channel. When a chemical messenger binds to the external binding site of the receptor, it induces a change in the protein shape, causing the lock gate to open up, allowing ions to pass through the ion channel. While in the resting state, the receptor is not bound to a messenger, so the ion channel stays closed. The binding of a neurotransmitter to ionotropic receptor results in a rapid response measured in matters of milliseconds. Typically, these are the receptors which fast neurotransmitter act. An example is the nicotinic acetylcholine receptor. Moving on to the second type and the largest family of receptors, which is G protein coupled receptors. These are also called metabotropic receptors or 7-transmembrane receptors. In general, they are activated by hormones and slow-acting neurotransmitters. These are membrane-bound proteins that are responsible of activating a protein called G protein. G protein is usually attached to the cell membrane inner surface, and it acts as a signal protein. When the chemical messenger binds to the receptor binding site, the receptor protein changes its shape, so the G protein can bind to it from the inner surface. When the receptor is activated, immediately G protein detaches from the membrane and binds to the receptor. This leads G protein to fragment into subunits alpha, beta, and gamma. These then interact with a membrane-bound enzyme and activate it. The active enzyme then works by activating a second messenger that leads to some intracellular effects, like calcium ion release or protein phosphorylation, and others, which all result in a cellular response. Not only that, a G protein can also activate or deactivate an ion channel, resulting in a change of the excitability. Therefore, a cellular response will result. An example belongs to this family is the muscarinic acetylcholine receptor, and it is considered a fast-acting receptor that takes seconds to produce an effect, but still slowest than the ligand-gated one. Type 3 is the kinase-linked receptor. These are a superfamily of receptors which activate enzymes directly and do not require a G protein or a second messenger. The receptor protein is embedded within the cell membrane, which parts of it exposed to the outer surface of the cell and parts exposed to the inner surface. The outer surface has the binding site for the chemical messengers and the drugs, while the inner surface has an active site. At resting state, this active site is closed, but when the messenger binds to the receptor, it causes a change of the receptor shape. This results in the active site being opened up, allowing the protein to act as an enzyme. So in this type of receptors, the protein itself has a dual function. It works as a receptor and enzyme. Once the enzyme is activated, it causes phosphorylation reactions by adding phosphate to the substrate. An enzyme that catalyzes a phosphorylation reaction is always called a kinase enzyme. So this receptor is referred to as the kinase-linked receptor. As long as the messenger molecule is bound to the receptor, phosphorylation reactions keep going. and this would result in an amplified response that direct gene transcription and protein production. Since the genes are involved in this function of the receptor, you would expect them to produce slower response in a range of hours. Receptors for insulin, hormone, and growth factors are typical examples. Lastly, in the most unique family of receptors is the nuclear receptors. At the beginning of this video, we said most of receptors are integral protein located in the cell membrane. Well, not all of them. This type is within the cell and are defined as intracellular receptors, or the nuclear ones. They are particularly important in directly regulating gene transcription. The chemical messenger for these receptors include steroid hormones, thyroid hormones, and retinoid. In all these cases, the messenger has to pass through the cell membrane in order to reach its receptor, so it has to be hydrophobic in nature. The response time is much slower than the membrane-bound receptor response, usually measured in hours or days. When the chemical messenger crosses cell membrane and seeks its binding site in the receptor, the binding causes a change of the receptor shape, followed by dimerization of the ligand receptor complex, so they form a dimer. The dimer then binds to DNA to induce gene transcription and therefore protein synthesis. This was an overview of these 4 types of receptors. Now let's see how our drugs can work on the receptors. Any drug that works on receptor can be either agonist, inverse agonist, or antagonist. In a case of agonist is when our drug binds to the receptor binding site where the endogenous chemical messenger usually binds and induces a cellular response. So there is an outcome. While the antagonist, it fits the binding site well, it binds to the receptor, but unfortunately fails to produce any response. So there will be no effect. So it just sits there and blocks the endogenous molecule from binding to its receptor. In the cartoon drawing here, chemical messenger like hormones and neurotransmitters, and the lock is the receptor itself. The key fits well in the lock and it opens the door, so there is a response. If we introduce an agonist to the body, although it's different from the real key, but it still can bind to the receptor and fits well there and successfully opens the door, so there is a response. While the antagonist, even if it fits the receptor, it fits the door, but it will never succeed in opening it, so there will be no response. Now with a key point summary of this video. Now we should know that most receptors are membrane-bound proteins that contain a binding site for hormones, neurotransmitter, and drugs. Binding triggers a series of events that produces a biological response. But as we said, not all of them are membrane-bound. They could be intracellular or nuclear. There are 4 main families of receptors that drug mostly target, and we have came across these 4. The drug acting on receptors can be either agonist or antagonist. And with this, we reached the end of this video. Thank you so much for listening. I hope it was helpful, and see you in the next one.